The effect of a particular gene depends on the environment. For most human traits, DNA does not contain a determinate genetic program from which we can predict a phenotype. The environment is as much a carrier of information as the DNA sequence, both for shaping phenotype and as a source of information in trying to predict phenotype. So what does this mean? It means you can't tell anything about a gene unless you know the environment in which it's being expressed. So let's talk about a single gene trait, such as freckles. So we have this trait, this phenotype that we're interested in, which is freckles. These traits are often influenced by multiple factors, but this particular one we know is influenced by the gene MC1R. So a person may inherit this gene, which gives them the ability to develop freckles, but only when the skin is exposed to sunlight. So what does the gene do? Well, it depends. If it's exposed to an environmental factor, then it will produce freckles. And what does the environment do? Well, it depends. It depends on if this gene, which allele is inherited. So this "it depends" statement is an example of a gene-environment interaction. They are both equally important in determining the phenotype. This also is an example of something called variable expression, and this is one of the reasons why Mendelian laws don't necessarily apply to human genetic traits, because you can inherit an allele of the MC1R gene, which allows you to develop freckles, but you'll have a variable expression depending on your exposure to sunlight. So even single gene traits have this variable penetrance or variable expression. Another gene-environment interaction. So you have a gene which has different alleles. The gene is the FADS2 gene. It's involved in fat metabolism. The trait that we're interested in is intelligence. So this gene is associated with an increase in intelligence as measured by IQ, which we are not going to get into the issues with that one. So let's pretend this is something that we can definitely prove. So this gene, a certain allele, is associated with a higher IQ, but only if a baby is fed an exclusively breast milk diet whenever they are young. So what is the impact of this gene, of this allele? Well, it depends on if the baby is breastfed or not. What is the impact on breast milk on IQ? Well, it depends on which allele you have. Another single gene disease, so PKU. So even one of these single gene diseases that we screen for in the newborn period that we know a lot about still can provide an example of a gene-environment interaction. We know the genotype, but the phenotype will depend on the environment, right? So PKU, so babies who have PKU and who eat Phi are usually healthy at first, but then symptoms appear within a few months. The symptoms may include slow growth, eczema, which is a skin rash, a musty body odor, small head size, and fair skin, because Phi is converted into skin pigment. Without treatment, children with PKU experience developmental delays, behavioral problems, intellectual disability, and seizures. Because early intervention can prevent the most serious symptoms, PKU is one of the genetic tests that we screen for in the newborn period. And with early intervention, many people with PKU have no health effects from their condition. So there's an allele that affects your ability to digest Phe. It's inherited in an autosomal recessive fashion, but what is the expression of this gene? Well, it depends. It depends on the diet. If you are fed Phe early on, then you will have the disease. What is the effect of the diet on brain development? Well, it depends on if you have this Phe, mutated Phe allele. Another example of a multifactorial disease. So now we're getting even more complicated. So you have a gene, the ALDH2 gene. This gene produces an enzyme that's important to alcohol metabolism. And there are two different variants of the gene, one that produces a functioning enzyme and one that produces a non-functioning enzyme. People who carry the non-functioning allele have a lot of problems metabolizing alcohol, and the broken enzyme causes aldehyde to build up in the body, and this is known as Flushing syndrome. It's relatively common, more common in certain areas, and so when an individual carries one of the variants and is heterozygous for this condition, they have one non-functioning and one normal allele, and they have a 100-fold reduction in alcohol metabolism. And what that means eventually is that they have a higher risk of esophageal cancer when exposed to moderate or heavy alcohol use, as compared to someone who has both functioning variants. And this particular type of cancer is pretty aggressive. So if you're heterozygous for this particular gene, so you have two different alleles that affect your ability to metabolize alcohol, and you are a heavy alcohol drinker, you will develop, this Flushing syndrome immediately, but also you have a much higher risk of developing esophageal cancer. However, now we're going backwards because your genotype can also affect your behavior, which can also affect your disease risk. So if you are homozygous for the non-functioning allele, so you are unable to metabolize alcohol at all, you actually have a decreased risk of developing esophageal cancer, because those people who are homozygous for these alleles get so sick when they drink alcohol that they avoid drinking alcohol, which decreases their risk. So one more example. The susceptibility to diseases are most often influenced by multiple genes and multiple environmental factors. So going quickly through this, please don't memorize this, but if we were talking about a disease like asthma that is very complex and very multifactorial, there are a couple of genes that are involved in metabolizing toxins that cause oxidative stress. And so you have a couple of different alleles, and you have some variations. We know that the risk of asthma increases about 50% if the individual carries a poorly functioning EPHX1 variant. The risk increases 4 times if a person carries both a poorly functioning EPHX1 and GST variant. So they're very poor at metabolizing the oxidative stress toxins. And children who have both high-risk variants and live close to a major roadway, because then they are exposed to those oxidative, reactive oxidant species. So children who have both high-risk variants and live close to a major roadway have a 9-fold increased risk of developing asthma. So this is an example of how genetic susceptibility to a disease can be exacerbated by environmental exposures.